Hi, my name is Chanel Lear, and I'm doing the extra credit project for Dr. Mukhtar's genetics class. So we're going to talk about replication, transcription, and translation. So we're going to begin with DNA replication. So DNA has to double prior to cell division. It does so in a semi-conservative manner, which means both parent strands are used as template strands. It moves in a 5' to 3' direction, and it occurs in 4 steps with the help of enzymes. So it goes through initiation, unwinding, elongation, and termination. So we're going to begin with initiation. To begin replication, proteins open up the double helix and prepare for complementary base pairing. The initiator protein binds to the origin of replication, which leads to the unwinding of DNA. Unwinding. Helicase unwinds the DNA by breaking hydrogen bonds between the two strands. Single-strand binding proteins keeps the DNA helix open and prevents the formation of secondary structures. DNA gyrase relieves stress on the DNA molecule by allowing free rotation and reducing torque. So now we're able to elongate. A primer made of RNA nucleotides is needed to help synthesize a new DNA chain. The primer is synthesized by primase and provides a free 3' hydroxyl group for the DNA polymerase. A primer is essential prior to the beginning of every Okazaki fragment. An Okazaki fragment is a short DNA fragment on the lagging strand and is discontinuous. DNA polymerase III produces a new strand of complementary DNA by adding nucleotides to the 3' end. DNA polymerase I replaces the RNA primer with DNA complementary to the template. Next, DNA ligase joins the Okazaki fragments together. It's now ready for termination, and that can occur in 2 ways. The first way is 2 replication forks meet, and the second way is telomeres produced by telomerase ends replication. So now we're ready for transcription. And here you can see this little animation of transcription happening, which is kind of cool to see it done this way. So now transcription, which is RNA polymerase transcribes DNA to produce an RNA transcript. So we're going from DNA and we're making RNA. A transcription unit comprised of a promoter, RNA coding region, and terminator must be present. A promoter recognizes the template DNA and start site. The RNA coding region is a sequence of DNA being copied into RNA, and the terminator is inside of transcription. So transcription occurs in 3 steps. We have initiation, elongation, and termination. So initiation, the transcription apparatus, which is comprised of RNA polymerase and proteins, binds to the promoter. When that happens, RNA polymerase can then bind to the promoter and begin transcription. RNA synthesis moves in the 5' to 3' direction on the template strand. The RNA polymerase unwinds DNA as the RNA strand grows, and we can see that happening in this picture. Next, we can terminate it, and we can do that by when the RNA polymerase transcribes the terminator, the transcription stops. And there's 2 types of terminators. We have rho dependent and rho independent. Rho dependent is the rho factor binding site and is usually A-rich. Then we have rho independent, which usually forms a hair loop and is C-rich. RNA polymerase is released at the terminator, and then RNA detaches from the RNA polymerase. So once we have our RNA made, RNA can go through some modifications. So we have the 5' cap, which adds guanine to the first nucleotide, which is the 5'. We also have the poly A tail, which is the tail of poly A's is added to the end, the 3' untranslated region. Then we have splicing, which takes out the introns, and then we have RNA editing, when you can add, delete, change the codon to code for different proteins. So we're following the central dogma. So we've gone from DNA to RNA, and now we want to produce proteins. And we can do that through translation. So translation in eukaryotes typically occur in the cytoplasm. Translation is the synthesis of a protein from mRNA. It occurs in 4 steps. The tRNA needs to charge, then you have initiation, elongation, and termination. So proteins are made of 20 amino acids, and then codons specify an amino acid in a protein, except for the stop codons. As you can see here in this picture, There are 64 different codons, 61 of them are sense codons, and 3 of them are nonsense codons. tRNA charging. So the tRNA binds to an amino acid on the 3' end, which you can see in this picture here. Recognition is based on size, charge, and R group of the amino acid. So once it is charged, we can begin initiation. The small ribosome subunit binds. Then the initiator tRNA binds to the mRNA. The large subunit binds and forms the initiator complex. The ribosome has 3 sites, the E, A, and P. Initiation starts in the P site. A charged tRNA comes and binds to the A site. The amino acids of each tRNA forms a peptide bond. The amino acid is released from the tRNA in the P site. The ribosome will then shift in the mRNA strand to the next codon. The tRNA in the P site shifts to the E site, and the A site shifts to the P site, so another tRNA can occupy the A site. The chain grows as new amino acids form and peptide bonds to the other amino acids. Now, once translation is complete, we can terminate it, and that is done when a terminator codon is translated. So the release factor binds to the A site, and the tRNA and mRNA can be released. So we can see that happening in this picture. And then there's this animation of translation happening, which is kind of cool. You can see them binding to the different sites, and you can see how the mRNA is just shifting. Thank you.